Good morning. Thank you to the Academy for inviting me, and many thanks to Professors Arnold and De Robertis for putting together the session. So I make it a little shorter. I will not mention much on oxygen. I will mention mostly on carbon dioxide levels and peroxide metabolism in mammalian cells. And at the end, since we are in a meeting about the Anthropocene, I will try to show how CO2 in the atmosphere can influence, potentially influence this type of biology. So just to give you a taste of the type of systems we work on trying to translate from basic human biochemistry to human disease and aging, I would like to show, for those that are not experts, these are redox reactions that occur in mammalian cells utilizing glucose as the substrate for energy production. So you have a first phase, which is the glycolysis, and then hydrocarbons, smaller carbohydrates get into this organelle, which is the energy hub of the cell, the mitochondria, where more than 99% of oxygen that we consume is utilized there, going all the way to water. So we call this the redox energy axis. Through oxidation reactions of glucose and metabolites, you lead to the formation of water and CO2. And through that process, energy released from the carbohydrates is stored in the form of ATP, which is our energetic coin. But at the same time, during these oxidation processes occurring in mitochondria, you generate a cocktail of oxidants all the time throughout oral life. And to show some of them, you can see superoxide radical anion, hydrogen peroxide, and peroxynitrite, just to mention a few of these reactive species that are formed all the time intramitochondrially, and even more in disease condition and during accelerating aging. So here you have a condition that in a microcompartment, such as mitochondria, which is the central hub of energy production in our cells, you have formation of water, formation of CO2, and usually CO2 has been seen as an inert, relatively inert molecule in human biochemistry. Just a few non-enzymatic reactions have been described with physiological significance. But still, there are lots of CO2 in mitochondria. There are lots of oxidants being produced in mitochondria all the time. So there is a perfect combination in the sense that that mixture, the CO2 reactions with the peroxides, may modify metabolism by fueling oxidation reactions. Yet, even though in the literature, chemical literature and biochemical literature, there was some scant information on the reactions of CO2 with peroxides, even from our group going back to the early 90s, very little attention has been paid to that interactions, much of that because it's very hard to measure these species in vivo since their half-lives are less than seconds, usually in the milliseconds or microsecond time scale. So it's really challenging to measure these transient intermediates, yet all the time they are being formed. So I like this sentence, this saying by Orwell. So in the redox field, we had this reality in mitochondria all the time, but we were trying not to look at it because it was just too complicated to take, a step forward. So people coming from the, CO2 production in biology, in the recent years, there have been great papers being published, CO2 being released by us. We exhale, all of us exhale 1 kilo of CO2 per day. So if we had 7 billion people living in the Earth, well, you can make an easy calculation and you can calculate how many kilograms of CO2 we humans are exhaling each day, 95% by our respiration, maybe 5% in skin, etc. So there is a lot of CO2 being released by humans and livestock, and it has been overlooked. So a few years ago, we put together a review article and then a second review article on this type of chemistries that we tried to go from the chemical aspects all the way to biology and human biology. So we call this the interplay of carbon dioxide and peroxide metabolism in mammalian cell. And this has to do also with redox switches that happens in the cell, connecting superoxide radicals also with nitric oxide, leading to the formation of peroxynitrite. So I'm not going to go into any details, but for those of you that are not experts on metabolism, this is the way biochemists look at intracellular reactions. And you can see here glucose which is the substrate we mostly utilize for energy production, going through a series of reactions of glycolysis to mitochondria in the form of pyruvate. And in that reaction sequence, you see acetyl-CoA and the Krebs cycle, and you see several sites where CO2 is being generated. This is why most of the CO2 we exhale come from our mitochondria. This is it. Of course, there is also CO2 formation in the cytosol by other pathways, and in parallel to CO2 formation, both in mitochondrial sites, you form peroxides, such as hydrogen peroxide, and extramitochondrial sites, you also generate peroxides, such as hydrogen peroxide and peroxynitrite. And peroxynitrite showed as the ONOO-, which is the diffusion control reaction between nitric oxide radicals and superoxide radicals. This is as fast as a reaction can go in biology. The second order rate constant is 10 to the 10th per molar per second. Extremely fast reactions occur biologically, very challenging to measure in cellular and even in vivo. So these are some early work done by the wonderful work done by Albert Lehninger's group at Johns Hopkins in the 80s, where they, in parallel, measure oxygen uptake and CO2 output by mitochondria. So oxygen consumption and CO2 formation are part of similar oxidation processes in vivo, although I'm not going to talk too much about the oxygen part because of time. So again, mitochondria being a central organelle for CO2 formation. So what are the levels of CO2? Because CO2 is converted to bicarbonate, right? So our blood contains 25 millimolar bicarbonate that comes from the hydration of CO2. So I made a scheme there. CO2 in mitochondria is about, in normal mitochondria, around 2 millimolar. And then that establishes a gradient with the cytosol and with the blood. And in the blood is 1.3 millimolar. And of course, this also affects pH, because the hydration reactions of CO2, when we generate CO2, leads to carbonic acid, and carbonic acid dissociates to bicarbonate and a proton. So if we generate more CO2, we tend to acidify ourselves. This is the situation. And these processes are highly accelerated biologically by a family of extremely super efficient enzymes called carbonic anhydrases. So in the last 20 years of research, and this is a consensus paper we did with authors all over the world, we were able to show that the product of the reaction of NO and superoxide, peroxynitrite, reacts fast with CO2 to form this adduct, nitroso-peroxocarboxylate. But what I want to show you is that CO2 activates peroxynitrite to generate radicals. So this is not radiation chemistry. This is just biology. We do form free radicals all the time. Some of them are NO2, which is nitrogen dioxide, the brown gas of the cities, and the carbonate radicals, which are CO2-derived radicals from our own metabolism. And of course, carbonate radicals are strong one-electron oxidants, can promote protein oxidation, including the oxidation of tyrosine residues, leading to the formation of tyrosyl radicals that then can evolve, for example, to oxidize or nitrated tyrosine residues in proteins. This is one of the oxidative post-translational modifications that we study in cellular and in vivo, and we know it is associated to aging and to disease. But CO2 also reacts slowly with hydrogen peroxide. Again, we go to mitochondria. There are two mechanisms by which CO2 can react with hydrogen peroxide to yield peroxomonocarbonate. Again, very tough to measure in vivo. We just released the first publication showing the cellular formation of peroxomonocarbonate, going from the chemistry to the biology. I'll show you later. So, peroxomonocarbonate is an activated form of hydrogen peroxide, because peroxomonocarbonate is about 100-fold more reactive than hydrogen peroxide. So when we talk about oxidative stress, there is no way we can neglect CO2, because CO2 is part of the process to generate 1 and 2 electron oxidants. So that goes back to the chemical literature, but to going from the chemistry to the biology, that's a big jump. And because these are short-lived species, that was the main challenge, and this is why these topics were somehow marginalized on the area. So now new publications are coming out. I'm finalizing take care of your cell culture systems when you use CO2, because CO2 with modulate signaling, this is a wonderful paper published by Elias Arner's group in Stockholm, showing that bicarbonate is essential for cellular signaling. And then we have done extensive work on the formation of the CO2-derived oxidants on the phagosome. What is the phagosome? The phagosome is the compartment by which macrophages and neutrophils engulf bacteria and parasites and kill them inside, dropping very strong oxidants, such as peroxomonocarbonate, you see the CO2 reaction with H22, and peroxynitrite. So these are CO2-derived oxidants that promote oxidative killing. And of course, if you are talking about CO2 reactions in the lung, as Professor Polsch was talking yesterday, you will see oxidative damage in the lung. So this is like a two-faced sword, because CO2 can be good or can be bad for oxidation reactions, depending on whether you want to kill the pathogen or if it's affecting your own host tissues. But CO2 not only influences directly redox reactions, but also through transcriptional responses. CO2 hypercapnia have to do also with transcriptional responses at the inflammatory gene levels. So oxidative stress and inflammation are intertwined, and CO2 is also intertwined with both. So we need to take care of that. And we really do not know, and I was talking about that with Eddie De Robertis, we do not know exactly which is the molecular CO2 sensor. There was a Nobel Prize for the oxygen sensor, which was HIF, but we do not know which is the actual CO2 sensor in biology so far. But we know there are transcriptional responses, we know it modulates inflammation, we know it modulates oxidative stress. To round up, the human age, the Anthropocene, what has this have to do with this? Well, we humans, the early Homo sapiens and the modern Homo sapiens evolved at 20% oxygen atmospheres and 0.042% CO2, which are about 250 ppms. Until when? For the last 300,000 years, no problem. But since 1950, CO2 levels are going up, and we are about double of the level of CO2 that we as species have seen over the whole evolution. And what happened 3 million years ago when the hominids started? Same levels. So hominids and the Homo sapiens and sapiens sapiens, we have seen CO2 at 250 for all our evolution, except for the last 70 years. Is that making any difference in our biology? We don't know that exactly, but I can speculate at the end of my talk a little bit. There is a wonderful paper published right before the pandemic started, the direct human health risk of increased atmospheric carbon dioxide. And this is not only an outdoor problem, but it is an indoor problem, because CO2 levels tend to go way up indoors because of lack of ventilation, high density of people, et cetera, et cetera, et cetera. Yes, if you go at levels above 1,000 ppm, then it starts to be a real problem. We are not so far from there, and serious, and really in indoor, we can be 2,000, 3,000, 4,000. This triggers inflammation, this triggers cognitive decline, this triggers oxidative stress. So some of these might be related to these reactions that I have discussed, the CO2 activation of the peroxide metabolism, and the aerosols that Professor Polsch was mentioning. I can tell you, in the aerosols of the city, H2O2 levels in a microdrop can be 70 millimolar. If that 70 millimolar hydrogen peroxide in the droplet is in touch with CO2, and this is now speculation from my side, that will affect your eyes, your skin, your epithelia, et cetera, et cetera. Just to show you the final slide, in the last 2 years, we were able to show that bicarbonate affects the glycolytic redox switch, affecting the glyceraldehyde 3-phosphate dehydrogenase step of glycolysis, turning off and turning on glycolysis. And we just published, together with a Brazilian group led by Ohara Augusto, the first report of the detection of peroxymonocarbonate in cellular. So this is the city of Montevideo, where we do much of our work. And again, thank you for your attention.